Okay, so we're just sort of picking up where we left off here. First, this slide here talks about the transfer of genetic information, how we get, information contained in the nucleus. How is that transferred within the cell, for example? So within the cell, the information, as you learned, is mirror copied and then transported outside of the nucleus, right? That's our cellular metabolic processes. The cellular metabolism is what we have that does that, right? It takes and builds things from information contained within the nucleus. From parent to progeny cells, right? So from the somatic cells of the parent to the cells that will, the sex cells, the genetic cells that will become the offspring. That's for tissue growth and repair. And then from parent organism to the offspring, genetic inheritance. Because we have genetic information, and because it can be transmitted to our offspring, survival at the cellular level is more guaranteed, right? It's easier to do because we have information in our cells. That information can be transported throughout the cells, throughout the life of the living organism, and then transported to the offspring, thus ensuring survival at the cellular level, the organism level, and the species level. And yeah, ionizing radiation, among other things, including random mutation. So I always like to remind people that random mutation happens too. Even if there was no radiation exposure, no natural background, no medical radiation, we would still have people developing cancer. We would still have people with genetic disorders, right? Random mutation would still be there. These things happening would just be at a slightly lower rate though, okay? But it's important to know that, right? That ionizing radiation, and especially medical ionizing radiation, can disrupt these processes. So it can disrupt the function of a cell within a person, the function of the tissue growth and repair of the cell, and then it can in principle disrupt genetic inheritance. Okay. The next thing we want to do is we want to talk briefly about the life cycle of a cell. Okay. A couple of important things here. Two broad categories to the life cycle of a cell. And again, the average lifespan is 24 hours, so think of this as a one-day thing, okay? But again, every cell, different cells are different. Some cells don't do mitosis, for example, right? Neurons, nerve cells, your brain cells don't do mitosis, right? So different cells do different things, but this is sort of the average way to think about it. So here you go. Mitosis is the phase of active division. That's what he showed in the video where he showed, the equator moment and the DNA splitting to either side. That was the end of mitosis. Mitosis is a very small part of the cell's life, that division process. Between mitotic phases, we call that interphase. So all of this rest of the pie that is not mitosis is referred to as interphase. Interphase is shown in that inner circle, taking up most of the pie. Okay. Mitosis is split into 4 phases. We'll discuss that briefly in a moment here. Interphase is split into phases as well. Interphase is the reproductive interim. Okay, it's what's happening, between cell reproduction. That's the cell's living its life. Okay, we call these 3 phases within interphase gap 1, synthesis, and gap 2. Obviously, maybe not obviously, but maybe you can tell me, what do you think's happening during synthesis? What do they mean? What's being synthesized? Yeah, the amino acids are being built into proteins, synthesizing proteins by the exposed genetic code. The cell's living its life, basically. The gap phases and synthesis phases are the cell living its life, building pieces for itself, running cellular functions, right? Doing its job, yeah? It's the life of the cell, okay? Where mitosis is the reproduction of the cell, right? And you know that, like, most of your life is living your life, right? And a very small part of your life is making new people, right? But most of the life is just doing normal things, going to work, eating, sleeping, right? Stuff like that. A small part of the life is making a new person, maybe. That's the point, right? Mitosis is a very small part of this process, right? The cell reproduces itself, right, through mitosis, and that's a small part of the story. Okay. So there we go. We'll touch more on that in just a moment here. Now we'll go back to our chromosomes and discuss more about structure of chromosomes. And actually, let me backtrack for just a second here. Let's look briefly at mitosis. Mitosis, if you read this in clockwise order, you have prophase, metaphase, anaphase, telophase. The 4 phases of mitosis and these different processes that are happening during that. So let's go to here. Between mitotic phases, you have what we call interphase. Now, during interphase, chromosomes appear only as a diffuse granular mass of loosely coiled chromatin. We'll define chromatin in a moment here within the nucleus. The point is, during interphase, the cell has to access the DNA. Okay? When the DNA is tightly packaged as a chromosome, it can't be accessed. The DNA has to be unzipped and unraveled in order to get to it, okay? But when it's unzipped and unraveled, interestingly, you can't see it under a microscope, okay? In order to see the DNA, you have to catch it when it's all packaged up during the mitosis phase, okay? So you can't see the DNA during the interphase. It's just not visible. It's too loosely packaged. Good. Chromatin is the name given to the string of DNA before it's like packaged up. Okay, series of bead-like globules of proteins called histones around which the DNA molecule wraps itself. This is interesting. Let me go to another slide and I'll come back to this. So, about to sneeze. Don't sneeze. Okay. All right. So the DNA is not just like sitting there and then just twisted up and stuck together in a big sausage thing. Okay. The DNA is interestingly wrapped around a protein periodically. So we have these proteins called histones. So this is one thing the cell has to build for itself, right? These little balls called histones are proteins, and these histones have, well, they're a protein. They're a string of amino acids that folded up into a little shape, okay? The cell has to build this. Once it builds it, it uses them to wrap the DNA around, okay? And you know, like if you're gonna, like, I don't know, have like a length of rope or whatever, it's usually better to have the rope like coiled up around something, right? Rather than just in a big ball, right? Because it tends to get tangled and things like that. So this is a way of organizing the DNA. This is one level of organization, right? So the first level of organization is, if you look up at the top left, there's the DNA double twisted ladder structure, right? Double helix twisted ladder structure. And then you follow it down, that twisted ladder structure wraps itself around proteins called histones, okay? And it'll wrap itself twice around a histone bead. So wrap itself twice around a histone bead, a little more strand, twice around the next histone bead, and so on, okay? That's called chromatin. So what we call chromatin is the strand of DNA wrapped around these histone beads, okay? These histone beads then all glob together, okay, to form something called the chromatin fiber. So it goes DNA double helix, wraps around histone beads to form chromatin. Those beads all gloop up together to form a sausage thing called chromatin fibers. Chromatin, the series of bead-like globules of protein around which DNA molecules wraps itself twice. It's interesting that it's twice. Like, it's not like 2 times around this one, 80 times around the next one, right? It's just twice around every one of them, and I bet you there's an interesting chemical answer to why it's twice, right? We're not going to go into that, but I bet you there's deeper answers, okay? Before extending to the next histone bead. All those histone beads with protein or with DNA wrapped around them, that forms our fiber. Strands of chromatin twisted together into a rope-like structure. It's sort of like if you look at any rope, any braided ropes, right? You'll notice that those braided ropes are made of smaller braids of rope. Those smaller braids of rope are usually made of individual little strands, right? Sort of the idea here, right? It's just different levels of organization to build a bigger thing, an organized bigger thing, okay?
If you wrap a rope around a ball, one time around the ball slip out. But
Sure.
if you wrap it at the top and the bottom, it holds it.
Yeah, exactly. So there's probably something similar to that as a reason why it has to be a couple of times, right? But what I think is interesting is why it's twice just exclusively, right? It's not like 2 times around this one, 6 times around this one, 3 times around the next one, right? It's just for whatever reason, it does it equally around every one of them. And there has to be an interesting chemical answer as to why that is the case.
And then what purpose does that have? Is it
It's just to give us different levels of organization, yeah? Because if you just took the DNA strand and then just smushed it all down, it's more likely that you'll have breaks and tangles and mismatches, right? But if you take the DNA strand, wrap it around something, and then glob all those somethings together, forming a longer thing, you can have more organized structure, yeah. I mean, that's the thing about the body, is that it just has all the way down organizational structure, right? All the way up to you're a full organism, seem pretty organized, right? And then down to the cellular level, we have our own organization, and then within the cell, it's organized further. The DNA gets its own nucleus. Inside the nucleus, the DNA organizes itself into chromosomes. The chromosomes organize themselves into strands. The strands are organized by, fibers, and so on, all the way down the line until you get to the individual genetic code. It has to be like this so that it can be reliably transported and reliably copied, and so it has to have levels of, we have to have levels of organization to be such complex organisms, right? Okay, good. And what I mean by us being complex organisms, I mean any living thing, anything that uses cells to live, the living unit, right? Good. Okay, so yeah, that's the deal, right? 3 levels of structure. DNA molecules wrap around histone beads, forming chromatin strands. Chromatin strands twist together, forming chromatin fibers. Chromatin fibers loop into shapes of 2 things called chromatids. So the chromatin strand becomes a long sausage called a chromatid. Okay, and just to be very clear here, you're looking at different levels of like zooming in, right? We are zoomed way in here, right? And we're zooming out and out and out through each of these things, right? Okay, so we're zooming, we have our DNA, we zoom out from the DNA, we notice the DNA is wrapped around some beads. We zoom out from the beads, notice all those beads are looped together into a sausage, okay? Or into a long fiber. We zoom out from that fiber, and notice the fiber is all packaged together to form that sausage thing called the chromatid. We zoom out from the chromatid, and notice that chromatid attaches to another chromatid, okay? And they attach in the center. The center attachment is called a centromere, okay? But this whole chromatid, what it really is just that, a big long string of that. Okay, all packaged down and organized. Okay. So yeah, here you go. A complete chromosome appears as 2 chromatid structures before synthesis phase, and as duplicated 4 chromatid structures after synthesis, because it has to be copied. Okay. Also, brief little history about the name here. What does some mean? I gave it to you guys earlier. Some means body, right? What's chromo mean? Color, yeah. So colored bodies, okay, is the name that these things have, okay? It sounds like an intelligent name, right? Chromosome, right? It sounds like a nice big intelligent name. Some smart scientists came up with that intelligent name, right? Here's how they came up with it, okay? The scientists Watson and Crick were looking through their microscopes under a special stage of the cell's process, right? And you can't see the DNA most of the time. They knew there's a nucleus, can't see the DNA. They happened to catch the DNA during the exact right time when it was all lined up into these little sausage-shaped bodies, okay? And you can't normally see them because the light just passes right through them, just like it passes right through everything else in the cell. But what you can do is you can inject some dye into the cell, and then if you inject the right kind of dye, the DNA will suck up the dye, okay? They happen to do that. They happen to inject the right kind of dye into the cell at the right time, looked under their microscope and noticed, there's all these little colored bodies, little colored things inside of the cell, okay? What are we going to call a colored thing? Well, chromosome, okay? Colored thing, colored body, right? They just looked at it, that's colored, right? And they go, all right, we're going to call it a chromosome, okay? That's where the name chromosome comes from, just because they looked at it and went, that's got some color to it. That absorbed the dye that we injected into it, right? That's where the name comes from. So, sounds intelligent, very intelligent people, but simple names from that, right? And that's sort of the story with a lot of things, right? It looks like we use our big big brains to come up with these things, but the names are usually derived from pretty simple ideas. Yeah. Okay. So good. That's a little bit about Watson and Crick. Okay. So next, we'll go over a little bit more of this cell life cycle. Okay. So mitosis is the phase of cell division. Interphase is the rest of the story. Okay. Now let's take a look at interphase. All right. Interphase has gap 1 synthesis and gap 2. During the gap 1, which actually takes up a big chunk of this pie, and on average of a 24-hour lifespan, this is 8 to 10 hours of the lifespan, organelles are duplicated during this process. Okay, so the cell's individual little organs, right, organelles, are going to be copied here. Okay, it's going to need to access the information in the genetic code to do that. So it's duplicating the organelles by accessing the genetic code, but importantly, it's not copying the full genetic, it's not making a copy of the DNA yet. It's using the DNA to build things with, okay? Library's open. We're gonna go into the library. We're gonna copy some books. We're gonna build things from that. That's gap 1. Synthesis phase, which happens after gap 1, around 6 to 8 hours on average, the cell replicates all the DNA. Synthesis means creation, right? Production. Cell replicates DNA, 2 chromatid structures become 4 chromatid structures for all chromosomes. So A goes to B. So we have organelles getting copied. Nextly, we have DNA getting copied. After synthesis comes gap 2, 4 to 6 hours of time. Enzymes and proteins are formed in preparation for cell division. Even though the chromosomes have doubled in their structure, they are still loosely coiled such that they remain difficult to observe under a microscope. So those are your 3 steps for interphase. Organelles getting copied in gap 1, DNA getting copied in gap 2, enzymes and proteins getting formed or built in gap 1, organelles copied, synthesis, DNA copied, gap 2, enzymes and proteins made. And all of that is being done by accessing the genetic code within the nucleus as in the same process that I described earlier. After interphase, we have mitosis, replication division. It results in full, identical complement of genetic material in each daughter cell. So you have a parent cell, and then you have daughter cells. We call them daughter cells because daughter cells can reproduce, and only females reproduce. So all cells made from this replication process are referred to as daughter cells. Mitosis gets its own 4 subdivisions, and we're going to talk briefly about those subdivisions. All right, here you go. Mitosis's subdivisions, okay? We start with prophase. During prophase, now remember, everything's been copied already, okay? So all we're doing now during these 4 phases of mitosis is basically putting things on the correct sides of the cell and getting ready for the rip apart, the split, okay? So during prophase, the nucleus swells to become much larger. It's going to take on more liquid, okay? Becoming larger. It's going to take up a big majority of the cell at this point. And then all of those loosely coiled chromatid fibers, chromosomes, are going to now, in phase B, are going to line themselves up along the equator of the nucleus, all in a row. Okay, that's what they showed in the video. Everything kind of sort of lining up in a row. He showed that complicated process of microtubules that pulls the whole thing apart, right? All the little walking molecules that run the process, right? Which I think is, I've watched that video I don't know how many times, and I'm always entertained by that, right? It's so neat to watch that whole process happening. But yeah, there you go. Metaphase, everything lines up. Okay, all the chromosomes. What I mean by everything is the DNA. The chromosomes line up in the nucleus. Okay, along the equator.
Okay.
Yeah.
Do they line up in a specific order?
Oh,
or
no.
Or is it just
Well,
completely random?
they very well could, but at our level of understanding, they don't need. There's no special order in which they line up. My intuition is no, that they don't, but I don't have a good answer for that. But that's a good kind of question to ask,
Sorry,
though.
you asked.
That'd be a good question to ask somebody who is, who had a much higher understanding of biology. Yeah, because they spend a lot of time in these biology. People who want to become a physician, things like that. They're gonna have a much better understanding of the actual micro workings here. But yeah, so they line up. We got 23 pairs of chromosomes. They line up. I don't know if they form a specific order in which they line up. That may very well be a good question. My intuition is no, but yeah. Importantly, though, for metaphase, this is the only time that the chromosomes are sufficiently densely packed where they can be visible under a microscope. This is the phase at which Watson and Crick saw their chromosomes, saw their little colored things, okay? Right here. Once the chromosomes are all lined up, and as the video I showed you said, the cell senses that it's ready, okay, it will then rip apart those copied chromosomes onto opposite sides of the cell. That's called anaphase. Anaphase, step C. Chromosomes are split and go to opposite sides. Telophase, we're on opposite sides now. Now there will form a nucleus around each separated DNA. Nucleus forms, and then the cell's membrane splits, like a bubble splitting into two bubbles. And then you have two new daughter cells because of that. Here's a photomicrograph of the process, okay? Prophase, metaphase, anaphase, telophase. Notice in telophase there's two new nuclei, and the cell is beginning to split down the middle. So, and this is just one little chunk of tissue in which they got lucky and were able to see all four phases happening in four different cells. Well, I guess five if you count the telophases too. But the point is that they were able to capture this for this picture in all four stages, which is really neat. Okay. How much time do I have?
There's more than one chromatid in each cell?
Yeah. You got 23 pairs of chromosomes, right? And each chromosome has two chromatids. Right. Yeah. So 46 chromosomes times 2, 80, 92 chromatid in each cell. Yeah. With the caveat that the the sex cells, chromosomes X and Y are going to be a little bit different. Like the X is the same, but the Y in males is going to be a slightly different size than the one for females. And that's an interesting thing, ladies. You actually have more genetic information than males do, okay? Because of your XX sex chromosome, you have more, literally a longer genetic strand than males do. The Y, a Y is less big than an X, right? If you draw a Y and an X there, the X has more like length to it, right? Because of that one little arm on the X. But yeah, so you actually have more genetic information than males do. Do whatever you want with that information, but you have more genetic information than males. We are. All right, so let's keep going. We're doing good so far, I think. So that was a brief description of mitosis. That's the division of somatic cells, the cells of our body. I want to just extremely briefly describe something called meiosis. Okay, meiosis is where a sex cell becomes a mature sex cell. So an immature sex cell, ovum or sperm, becomes a mature one. Okay, all cells start out with 23 pairs of chromosomes, but ova and sperm have to have only 23 chromosomes. They have to have half the number of a somatic cell in order to be used. So there's a process called meiosis, which if you think about mitosis is equal division, right? Everything's copied, everything's split, right? You can think of meiosis as an unequal division. Everything splits, but not equally, Okay? It's characterized by replication division followed by reduction division. So you have a mitosis process, but it is followed by a reduction process, a way to reduce the number of chromosomes. Replication division is all 46 chromosomes are passed on to 2 new daughter cells, where reduction division is only half of the chromosomes from the daughter cells are passed on to new daughter cells, which again have only half. Okay. So in reduction division, synthesis is skipped. Remember, synthesis is when the DNA is copied. Synthesis phase is skipped once. So let me show. You have a immature cell that will eventually become a sex cell. Okay, 46 chromosomes. The first step for this is to make 2 daughter cells from it. Those daughter cells will be identical. Okay. The second step is split the cell again, split each cell again, but when you split it, do everything except for copy the genetic code. Okay? No copy of the code means you just rip the code in half. Okay? And give half of it to each cell. So the reduction division skips synthesis phase during interphase. I know there's a lot of phases, but during interphase, synthesis gets skipped, and the outcome is from one initial cell, 4 daughter cells that have only half of the number of chromosomes. You notice it says 23 for the total number here. So that's the deal. Synthesis gets skipped one time after the first division, and you have now cells that can. There's a lot more to the story about how the ova and sperm actually develop into mature ova and sperm, but this is the story for how they obtain the right number of chromosomes to be used in sexual reproduction. Okay. All right, we've made it through, thankfully, through the part of the review of biology that we need to understand how radiation affects the body now. Okay, so now for the rest of this, as much time as I have for today, I get to talk about how cells are now sensitive in different ways. We're done with our biology review, thankfully, so let's move on.